Hi everyone, I am Dr. Waseem Sajad, and today we will try to learn how we can predict the age of the hematoma or hemorrhage inside the human body. And for this purpose, we will use MRI sequences. MRI sequences help us to predict accurately the age of the hematoma or age of the blood, because as the blood degrades into its forming products, the signal on MRI changes. With the help of these changes in MRI signals, we can predict whether this hemorrhage or hematoma is acute, hyperacute, subacute, late subacute, or chronic. So first of all, we try to learn what happens when a blood comes out of the blood vessels. When the blood cells come out of a blood vessel, first of all, the blood cells are intact. Then, with the passage of time, there is hypoxia of the blood cells, and with the more passage of time, this hypoxia becomes severe, and eventually there is lysis of the blood cells. Meanwhile, there are changes in the hemoglobin in the red blood cells. First of all, there is intracellular oxyhemoglobin, then there is intracellular deoxyhemoglobin, then there is intracellular methemoglobin. When the lysis occurs, there is extracellular methemoglobin, and eventually there is ferritin or hemosiderin. So these blood products basically are the reason of difference in the MRI signals at different ages of the hematoma or the hemorrhage. So we need to learn at what point of time what is the status of red blood cells and the status of hemoglobin. Then corresponding to this status of the hemoglobin and the blood cells, what will be the signals on MRI sequences. For MRI sequences, T1 and T2 images both are valuable and both are mandatory to predict the age of the hematoma. Neither T1 alone nor T2 alone can predict the age of the hematoma or the cerebral hemorrhage. So we use T1 sequences and T2 sequences, compare these two sequences, and then predict what is the possible age of the hematoma and the cerebral hemorrhage. So we divide the hematoma or the cerebral hemorrhage into different types depending upon the age. There is hyperacute form, there is acute form, there is early subacute form, there is late subacute form, and there is chronic form. In hyperacute form, the hemorrhage has been occurred in less than 12 hours. It means we label a hemorrhage or hematoma hyperacute if the time from extravasation of the blood from the blood vessels into the surrounding parenchyma is less than 12 hours. If the time is from 12 hours to few hours, then we label it as acute, and when it goes from hours to days, then we label it as early subacute. When few days has passed, we label it as late subacute, and after 30 days we label it as chronic. So in hyperacute form, there is just the extravasation of the blood from the blood vessel into the surrounding tissue. The blood cells are intact, and the hemoglobin inside the blood cells is oxyhemoglobin. it means it is oxygenated hemoglobin, and the signals on the T1 is low and the T2 is high. It is hyperintense on T2 and hypointense on T1. That is the hyperacute form of the cerebral hemorrhage or hematoma. As the time passes, there is hypoxia of the blood cells, there is deoxygenation of the hemoglobin. The hemoglobin is deoxygenated, but it is still present inside the red blood cells because the red blood cells are still intact, and the signals on the T1 weighted images is either isointense or hypointense, and the signal on the T2 images is also hypointense because the oxygenation of the hemoglobin is not now present. It is deoxygenated hemoglobin and it appears hypointense on T2 weighted images. Now, when the more time passes, there is early subacute form. In early subacute form, there is hypoxia of the red blood cells, but the red blood cells are still intact. The hemoglobin changes from deoxygenated hemoglobin to methemoglobin, and the signals on the T1 weighted images now is hyperintense, and the signal on T2 weighted images is still hypointense because it is methemoglobin, it is not the oxygenated hemoglobin, and the signals on T2 is hypointense, and the signals on the T1 weighted is now hyperintense. In the late subacute form, there is lysis of the red cells, and now the methemoglobin, due to lysis of the red blood cells, is now extracellular. It is extracellular methemoglobin, and the signals on the T1 weighted images is hyperintense, and the signals on T2 weighted images is also hyperintense due to lysis. Now the next form is chronic form after 30 days. Now this methemoglobin further degrades into ferritin or hemosiderin, and hemosiderin and ferritin on both T1 and T2 sequences show hypointense signals and appears dark. So with the help of these MRI sequences, we try to predict the age of the hemorrhagic stroke or hematoma. Now there are different types of acronyms and different types of mnemonics which are used to memorize the different sequence of T1 and T2 on different stages of the cerebral hemorrhage. But my advice is you will forget these mnemonics and these acronyms. The best way is you need to know what is the state of the blood cells and what is the status of the hemoglobin at that particular time and the particular stage of the cerebral hemorrhage and hematoma. And then when you know what is the status of the hemoglobin and the red blood cells, then with the help of this status try to predict the T1 signals and T2 signals. If we repeat these signals in hyperacute form, only T2 is hyperintense, T1 is hypointense or isointense. In acute form, the T1 is still hypointense or isointense, and now the T2 sequence is also hypointense. In subacute forms, in early subacute and late subacute, the T1 sequence is hyperintense. T1 sequence is hyperintense only in early subacute and late subacute. Now, in early subacute, there is intracellular deoxygenated hemoglobin and methemoglobin, and the signals on the T2 is hypointense. In late subacute, there is lysis. Due to the lysis, the signals on the T2 is hyperintense, and in chronic forms, there is hemosiderin, and the signals in both T1 and T2 is hypointense or dark signals. So, I hope that with the help of this understanding of the status of the hemoglobin and the status of the red blood cell, we are able to predict the signals on the different sequences of MRI, and with the help of these sequences, we can predict whether the hematoma is hyperacute, acute, early subacute, late subacute, or chronic. Thank you.